Hi, today we'll show you how electroelution is performed. In order to purify DNA fragments, such as digestion and PCR products, for further purposes like cloning and sequencing, electroelution can be one of the methods of choice. Based on the negative electric charge of DNA, a high salt barrier is placed within the electrophoretic field, trapping the fragments and preventing their further migration. This method allows the purification of a wide range of DNA fragment sizes from different sources, with a relatively high efficiency of up to 80%. Its low costs represent another advantage of electroelution when compared to other purification methods, such as columns, resins, kits, among others. The DNA sample is first visualized by a titanium bromide stained agarose gel electrophoresis, in order to visualize and identify the desired fragment to be purified. Electrophoresis should proceed until fragments are well separated and their size can be precisely determined. Following electrophoresis, the desired fragment is neatly excised from the gel using a brand new razor blade and taking care to trim the gel slice as close as possible to the band of interest. This procedure should be performed with a proper protection from UV light and wearing gloves. After filling the electrolyzer with the appropriate burning buffer and taking care not to spill it over the surface of the middle plateau, the gel slice is placed inside one of the wells, as close as possible to the V channel. There should be no bubbles inside the V channel. This can be avoided by flushing the channel with buffer to remove any trapped air. Then, 100 microliters of the high salt barrier are added carefully to the V channel from the side proximal to the well. The chamber is then closed and the electrodes connected to the power source to begin electroelution at 100 to 120 volts. The time lapse will depend on the fragment size. For large fragments of up to 20 kbs, 50 minutes to 1 hour should be enough, and small fragments should be monitored every 10 minutes with UV light to avoid further elution through the salt solution and into the anodal buffer chamber. In this case, we digested 560 nanograms of plasmid with the enzymes NDU1 and HIN3 to release a 900 base per fragment, equivalent to 84 nanograms of the sample. Accordingly, after placing the gel slice inside the well, electroelution will proceed for 25 minutes at 100 volts. Once electroelution is completed and DNA fragments are trapped in the salt cushion within the V channel, 400 microliters of solution are recovered from the channel from the distal side of the receptacle. And placed in a 1.5 microliter microfuge tube using a fine tip attached to a micropipette. Proceed to alcohol precipitation by adding 2 volumes of cold absolute ethanol and mix by vortex. Additionally, 2 to 3 microliters of glycogen may be added to improve the yield of DNA recovery. Place the tubes at -20 degrees Celsius for 1 hour, or alternatively, store at this temperature overnight. Centrifuge at 4 degrees Celsius at top speed for 25 minutes. Discard the supernatant and wash by adding 1 ml of cold 70% ethanol. Centrifuge at 4 degrees Celsius at top speed for 5 to 10 minutes. Discard supernatant and allow to dry at room temperature with the tubes in an inverted position. Resuspend in 10 to 15 microliters of nucleus-free water and proceed to determine the concentration of the sample. case, the purified sample was resuspended in 10 microliters of water and quantified in a spectrophotometer at 260 nanometers wavelength, showing a concentration of 6.3 nanograms per microliter, representing a 75% efficiency. You may be thinking you'd be better off investing only 10 minutes of your precious time with a kit. However, in spite of the time consumed in this process, you will be able to control many of the variables that will allow you to purify a wider range of samples, varying in nature, sizes, and sources. As you could have seen, in the present case, a 75% efficiency was observed, equal to or even better than most DNA purifying kits. Thank you for joining us today. We hope that you have found this protocol useful for your research projects.